Okay, hey biochem. So I was hoping that my voice would be back by now, but it's not. So I'm gonna try to make this a really short video. I have a whole bunch of learning objectives. Maybe you can just read them on your own. I will point out one. Just draw the mechanism for the reaction of lipase. You've already done this before. You've already drawn this one, so I would suggest you look back at your exams and previous assignments for this one. But today is the first day of fatty acid. FA is abbreviation for fatty acid catabolism. Okay, so here's the outline. So we're going to have 3 days of this. These first 2 are going to be day 1, which is today, and this is going to be day 2. So we're going to do lipolysis, activation, and transport. And then day 2, we'll do beta oxidation. And then day 3, which I didn't include here because it's not exactly part of a flow, we're going to do nuances and diseases. Okay. All right, let's get started. Okay, so let's imagine the scenario is that you eat a stick of butter. I know it's kind of gross. And then go run a marathon. Okay, so we're imagining that your glycogen is being depleted and that the energy that you need to sustain and to make ATP so your muscles can keep moving and you can keep breathing and your brain can keep working is going to come from metabolism of fat. Okay, so instead of storing it, you're going to be using it. So the first thing that happens is you eat the butter, and it's mostly TAG, the triacylglycerol, and that crosses through the intestinal mucosa. And in order to be transported or to get where it needs to go, because it's not soluble in the blood, it has to be packaged. So it gets packaged into what's called a lipoprotein particle. Okay, so here's a vocab word, lipoprotein particle. And the specific lipoprotein particle, there are 4 lipoprotein particles. Of course. Okay, hold on. Okay. Those fluffs have been crazy all day. Actually, all weekend, really. Okay, so the tag, here's an example of a tag. Because remember, it has tri means 3. It has 3 acyl tails. It will get packaged with some cholesterol. That's cholesterol right there. Into a lipoprotein particle. And a lipoprotein particle is exactly what it sounds like. It's part lipid, part protein. The outside is protein. That's what makes it water soluble. Or in other words, enabled to be transported through the bloodstream. These tags that you eat are packaged into a very specific lipoprotein particle called a chylomicron. And this is one of the 4 lipoprotein particles that you'll need to know. The other ones are over here. So I picked this figure because it shows relative size. So you'll need to be able to rank these in terms of size, but then also describe their function. So the way that I like to think about these lipoprotein particles is that, for example, this is a chylomicron up here, the really big giant one. It transports tags. It's kind of like a FedEx jumbo jet. It's just primary function is to get packages from one place to another, but it's so humongous that I would compare it to like a jumbo jet airplane. Whereas VLDL also transports tags, but maybe it's more like a FedEx van. And then these might be the LDLs, or the HDLs and LDLs might be like little drones. So all the protein particles, are just for transporting. They just carry lipids and drop them off and pick them up. So we're just going to go through them from smallest to largest. Okay, the smallest obviously is HDL, which stands for high density lipoprotein particle, and its main function is to remove excess cholesterol. And it brings it, when it removes it, it brings it back to the liver. And then you have LDL, which is low density lipoprotein particle, and that brings cholesterol from the liver to wherever it needs to go. So it brings or delivers cholesterol to anywhere that it needs it. We'll just say where needed, like an arterial cell wall, it would transport that. So what you'll notice is that the HDLs and the LDLs are cholesterol transporters. So that's the lipid that they transport. Whereas VL is very low density lipoprotein particle, and the chylomicron these transport tags. VLDL transport tags from the liver. So if it's synthesized, made in the liver, then it gets sent out, and chylomicron takes them from where you've eaten them from your intestinal mucosa and takes them to where they need to go. So those are the lipoprotein particles. Again, they're just transporters of lipids, and they transport specific lipids, and they're of specific sizes. And I've included what I think is a really interesting video, obviously, hopefully, well, yeah, not narrated by me, thank God, that will relate a little bit into disease. Okay, so now the butter that you've eaten is a tag, primarily. It's gone from your stomach to your intestinal mucosa to a chylomicron and is now being transported to the blood. And it's going to be dropped off at a cell. In this case, this particular figure is an endothelial cell. When the chylomicron is recognized by this protein, that's an enzyme called lipoprotein lipase, also known as just lipase. So, as you can see, the lipase recognizes the chylomicron, and then the enzymatic function is to take the tag, the triacylglycerol, and break it into the individual acyl tails or fatty acids and glycerol. I'm going to sneeze. Okay. So the overall equation for this is you'll have a tag. I'll just move this over to the side. Triacylglycerol, please don't forget how to draw these. The glycerol is the 3-carbon backbone. Each carbon gets an oxygen. Oftentimes, this acyl tail is abbreviated with an R. And just as a reminder, this functional group right here is called acyl. It's an acyl group where the carbons coming off is more than one. If you remember, the CH3O, R is an acetyl group because there's only one carbon. So acyl means there's more than one carbon coming off of here. Okay. No, that's in my way. Hey, you gotta go away. Sorry. Acetyl group. Okay, so you take a tag And you're going to need 3 waters, 1 to cleave each tail. And this is catalyzed by the lipase enzyme. And what you get out is glycerol, and 3 fatty acids. Okay, this is the mechanism that you already know. Oh, I guess I've got to say it. Sometimes, see that little goofy thing right there? Sometimes you'll also get a monoacylglycerol coming out. I don't know why this is important, but it turns out to come up on a lot of questions. Sometimes, so you might get a little of monoacyl. Okay, so just to remind you, this is the active site. For lipase. And you'll notice it has a serine, a histidine, and aspartic acid. So the mechanism is analogous to a protease. Only in this case, because it's a lipase, we cleave a lipid instead of a protease cleaves a protein. So again, this is the mechanism that you've already done several times before. So I would strongly suggest that you review it. Okay, now we have our glycerol and our fatty acids inside the cell. And the glycerol, if you'll notice, has 3 carbons. And the pathway that we know that has a lot of 3-carbon intermediates is called glycolysis. So glycerol will eventually get transformed into this molecule called glyceraldehyde 3-phosphate. That part's not particularly important. But I just wanted to show you that the carbons from glycerol are not wasted because we have a lot of carbon hydrogens. We can get some anaerobic ATP from the glycerol that came from the tag. So just so you know, those 3 carbons from glycerol are not wasted. They're converted. You put a phosphate on it, do a little oxidation, and then that 3-carbon intermediate can enter into glycolysis, which can go to PDC, which can go to the TCA cycle. So I just wanted to mention that. Okay, so we have the free fatty acids are in the cell, so what happens next? So what I'm going to do is I'm going to draw a really big overview, and we're going to come back to this giant molecule here. I just didn't know where else to put it. I'm going to make this really tiny. Okay, so it's going to be a recap of metabolism thus far. This is the cytoplasmic membrane. This is the outer membrane of the mitochondria. This is the inner membrane, inner, I-N-N-E-R. This is the matrix of the mitochondria, and this is the inter with a T, membrane space. And then this is the cytosol. And again, we make this the blood. Okay, so it's just as a recap. If you have glucose that comes from glycogen or your diet, enters through a permease and then undergoes glycolysis to pyruvate. Get 2 of those. We get a little bit of the pyruvate, undergoes fermentation to lactic acid, and we get out is So some of the pyruvate will undergo fermentation to regenerate the NAD+ for glycolysis to keep the aerobic metabolism going. But a lot of the pyruvate will enter into the mitochondrial matrix and will be oxidized to acetyl-CoA. Get some CO2 out here. And then the acetyl-CoA from the PDC, pyruvate dehydrogenase complex. Also, sometimes we call this the bridge step. The acetyl-CoA will go into the TCA cycle. And what I've done, and I just have one turn for each of these, one spin. And this is where you get the final two CO2s coming out. And then we get a bunch of reduced cofactors, coenzymes. Okay, that's just a reminder of carbohydrate catabolism or oxidation of a glucose, and the reason that I'm doing that is because we're going to need to connect them eventually. Okay, so let's say we have a chylomicron, and inside of it are a bunch of tags. So these look like E's, but these are my tags. Okay. So it comes up to a cell and is recognized by the lipase enzyme, and that lipase enzyme will break apart these two into glycerol and 3 fatty acids. I'm just going to do Fa. So glycerol is a 3-carbon molecule, and it will enter in sideways, not at the beginning, obviously, because glucose is 6 carbons. But after glucose is broken apart into the 3-carbon intermediates, glycerol can enter in a little less than, well, no, approximately halfway through the pathway. Okay, so we have our 3 fatty acids are inside the cell. Now I'm going to focus on a number of membrane-bound enzymes and transporters. So I'm going to zoom in so that we can get as much out of this as possible. And I'm going to draw them in different shapes just so that you understand that these are all different enzymes. Obviously, like this first one is not heart-shaped, but it's just a shape that was different from the rest of them. Okay, so this is lipase does lipolysis. That's the first part of fatty acid oxidation is lipolysis. The second part, so maybe I'll do like this, is called, go back, is called lipolysis or break the fat. And then the next part is called activation. So the fatty acid is activated to become an acyl-CoA. So I'm going to draw some of this out. So remember fatty acid. Okay, so this next part here is going to be called activation. And the reason we have to activate the fatty acid is because any fatty acid that's more than 12 carbons long cannot get into the matrix. And if it can't get into the matrix, then you can't break down a fat. And then that's not good because you get lots and lots of energy out of a fat. Okay, so you notice that we created a thioester. So I need to add a CoA. So a CoA goes in. And hopefully you remember that that's going to cost a ton of energy. And the source of that energy is coming from an ATP. Now watch this part because it's really unique. This is the only time you're going to see this. The ATP is hydrolyzed to That's monophosphate. And pyrophosphate. So PPI is pyrophosphate. So, and then these two can be broken apart into two inorganic phosphates. So you get a lot of energy is released when you cleave off both of the inorganic phosphates off of the end of ATP. And this is catalyzed by this heart-shaped enzyme. It's not really heart-shaped. Please don't think it's heart-shaped. I just picked a shape. It's called acyl-CoA synthetase. There's a T in there. Please don't forget about that T. Okay, so that whole process where a CoA gets added on, and the cost to add the CoA on comes from breaking down an ATP to an AMP, is catalyzed by acyl-CoA synthetase, and that gives us an acyl-CoA. So this whole thing right here is called an acyl-CoA. Okay, so it turns out this acyl-CoA cannot get across the membrane itself. So we have another enzyme. I'm just giving it a different shape, just so that you understand that there's different enzymes here. And this one is going to take the acyl-CoA, and we're going to take off the CoA, and we're going to add a molecule called carnitine. And I didn't leave enough space for that. Okay, so we're just going to flip-flop, swap them out. So this is now going to be an acyl-carnitine. Okay, now acyl-carnitines can get across the first membrane, no problem. Why is it not letting me erase that? There it goes. Okay. Okay, so acyl-carnitine can get across the outer membrane, no problem, and then it has a very specific transporter that recognizes the carnitine plus the acyl group. And once it gets through, the carnitine is again swapped back out for a CoA. So we get the acyl-CoA back out. So again, the CoA goes in and a carnitine comes out. All right. Let's see. This process right here, where we swap out the carnitine, the acyl carnitine crosses, and then we swap the carnitine back out for the CoA, is called the carnitine. Wow, you can't read that at all. Let's try again. Carnitine shuttle. And it's the rate limiting step. I should maybe call it the rate limiting process of fatty acid oxidation. Okay, I'm going to go through this one more time because we have three things going on. We have, this is lipolysis, then we have activation, and then this whole thing in yellow here is transport. Okay, so a chylomicron arrives at a cell that needs energy from a fat because it's we're starting to run out of our carbohydrate energy. A lipase enzyme will break a tag apart into glycerol and 3 fatty acids. The glycerol carbons are not wasted. Those go up to glycolysis. And then on to the PDC and the TCA to get some energy. The fatty acid has to then be activated. And it's activated by getting a CoA to become acyl-CoA. And in order to put the CoA on, ATP is broken down into AMP and pyrophosphate, and that enzyme is called acyl-CoA synthetase. Then the acyl-CoA, which cannot also cannot be transported, the CoA is swapped out for a carnitine molecule, and I'll show you what that looks like in a second. And the acyl carnitine can transport across. And then on the inside, on the inner mitochondrial membrane, the carnitine is swapped back out for the CoA. And that process is the transport of the acyl group into the matrix. So if you'll notice, what color should I use here? This acyl group is this acyl group. Acyl is this acyl group. Is this acyl group. So we've got the carbons moving from the cytosol into the matrix. And the carnitine shuttle is the rate limiting step. One other thing that I want to point out that's going to become important later is that you'll notice that you have CoA in the cytosol and that you have CoA in the matrix. And these are not the same CoAs. So the cytosol has a pool of CoA molecules, and the matrix does as well. But they're not the same CoA. So if we were to radio label them or make them all carbon 13s, you'd notice that these CoAs never actually cross the membrane, and they're different from those CoAs, and that's a regulatory aspect of all of this. Okay, so I need to show you what carnitine looks like, because you'll just need to be able to recognize this molecule. So this is carnitine. It has a functional group that you've seen before called a choline. And then it has a carboxylic acid and an OH. And carnitine is in a lot, if you go look online, it's in a lot of weight loss supplements. And the claim is that if you increase the amount of carnitine that you eat, then I've got so many colors going on here. I want to keep drawing on this, but I can't. If you increase the amount of carnitine that you eat, then you should be able to increase the amount of fat that gets transported into the mitochondrial matrix. And if a fat gets transported in the mitochondrial matrix, it has to be broken down. It's not coming back out. That's true. So if you increase one of the molecules needed in a rate-limiting step, you should increase your fat breakdown, i.e. your weight loss. So that's why carnitine's in a lot of weight loss supplements. And I think, okay, that's all that I have. I have no idea how long this ended up being. Hopefully you can understand it, and I'm hoping that I'll get my voice back really soon so the rest of them won't be as rough as this one. I'll see you in class. Bye.